staying with these bacteria that produce antibiotics, and went on to study how they divide. They look superficially like bread mold, like a fungus. So they grow not as single cells, but as a branching network of hyphae. And the spores are the way the bacteria can sort of disseminate themselves in the environment. What usually happens in unicellular bacteria, which most people are familiar with, is that the cell grows, as the cell grows larger, then it divides in the middle. These Streptomyces, which grow as extended networks of hyphae, they don't need to do that. We could interrogate, if you like, the process of cell division by creating mutants of these bacteria, which could no longer sporulate, produce the spores, but they would still be viable. Now, if you do the same kind of interrogation in a unicellular bacterium, the bacteria just die because they can't divide. So we had a model system, if you like, to study cell division. And we've done quite a lot of nice work on understanding how the machinery of bacterial cell division works. I'll just finish off by talking about Streptomyces and antibiotics, because that's really why we study them, because they produce all these useful compounds for us. In 2002, the first complete genome sequence of one of these bacteria was produced and published in Nature, the journal called Nature. We discovered there were genes which would enable the bacteria to produce an even more diverse set of antibiotics, so upwards of about 15 to 20 different molecules. This opened the door, effectively. This was just the tip of the iceberg, because as other members of the genus, this group of bacteria called Streptomyces, as other members, other species were sequenced, their genomes were sequenced, it became a rule, effectively, that there were what are called cryptic biosynthetic pathways for producing antibiotics. So the bacteria have these pathways, the genes, which would enable them to produce other antibiotics, which we didn't know about. So we wanted to know about how to trick the bacteria into using these pathways. And it's that which I'm currently working on with, in a collaboration with a group in China. And so we're really near the beginning of this investigation, but it does seem very, very exciting. As you may know, there's a worldwide crisis in antimicrobial resistance. There's a really urgent need to discover new antibiotics with which to combat these resistant pathogenic bacteria. So that's why I'm really excited by this discovery of how we can trick the antibiotic producing bacteria to produce these unknown products, these new antibiotics, which I think will be very helpful, very useful in our ongoing race against resistance in the disease-causing bacteria.